Hi everyone, welcome back. This video is a continuation of what we have done in the last tutorial where we looked at the dataset from Visium from 10x and performed single-cell RNA-seq analysis. Well, we did some basic analysis where we did pre-processing, we did differential analysis, and then we actually performed some clustering to identify various clusters in our dimensionally reduced dimensions, I should say, or dimensionally reduced data set, and we left it there actually. So we identified these clusters as 0, 1, 2, 3, 4, 5, 6, and so on, because how many ever clusters we had, we labeled them as that many, right? Now, the goal for this tutorial is to take it to the next level just by assigning a cell type for each of these clusters. For example, we have a cluster, and that cluster comes with a gene profile, right? So we're going to match that gene profile against a database and say, okay, with so much score, with so much probability, if you want to call that, I'm going to call this a certain type of cell. It could be B cell or a T cell, and so on. So that's the goal for this exercise. And again, we're not going to identify the tissue types. I'll briefly explain the difference between cell type and tissue type for those of you newbies to this bio field, because these may sound similar, but obviously slightly different terminology. Okay, and if you want to be notified of any future similar videos, again, it makes sense for you to hit the subscribe button down below. And while you're there feeling extra generous, try to find the little thanks button. And now let's go ahead and jump into just a few slides before we jump into the code. First thing first, a cell type annotation. That's the title of my, of what we're trying to do, but what exactly does it mean? I kind of quickly summarized it, right? This is where we left it off last time. We had these clusters and we labeled them 0 to 12. We had 13 clusters identified here, and we kind of mapped them onto the original tissue to see where the locations are. Now, based on this, you can kind of do multiple things. You can label these areas. If you know exactly what these areas are, you can label them based on these spatial locations. You can label them based on tissue types. You can label them based on cell types, which is what we're going to focus on right now. So cell type annotation is the process of assigning or identifying these specific cell types. How are we doing that? By looking at the gene expression patterns. Okay, things make sense hopefully once we go through a couple more slides and then jump into the data. So here is a quick snapshot. Too much text. I hate slides like these, but it helps you freeze the screen and then kind of quickly read through it. But let's go through it. Cell type identification. Obviously, we are identifying individual cells based on their unique characteristics. And when it comes to tissue type, it's a broad category or a classification of a specific type of tissue. You have a tissue and it's a specific classification. And how do you define a cell type? You define it by looking at the molecular, phenotypic, or functional attributes of a individual cells. And very similarly, a tissue is basically how the cells are organized, the composition and the function of the collection of these cells in that specific tissue. The examples probably may clear things up. On the left-hand side, the examples include, for example, neurons are a specific cell type. T cells, these are the type of white blood cells, right? They are a specific type of cell. Cardiomyocytes, these are, as the name suggests, cardio, they have to do something with the muscle. So wherever a muscular type of action is required, then you can find these cardiomyocytes, right? And fibroblasts and lymphocytes. These are specific type of cells. But if you are looking for tissues, then there is an epithelial tissue, muscle tissue. So in a muscle tissue, you may find cardiomyocytes. Hopefully that makes sense, right? In epithelial tissue, you may find T cells, for example. So this is, and for example, nervous tissue, right? In a nervous tissue, you may find neurons. So there is hopefully you can find the link right there. And the cell types are typically defined by a specific gene expression pattern, which is what we have right now. So based on the gene expression pattern, we're like, okay, this is this type of cell. And the tissue types are usually defined by the architecture, the cellular architecture and their histology. And it kind of requires a little bit of bio knowledge, I should say, although most of that is being automated. For example, cell types, you can actually match your patterns, the one that you get, against some database by applying certain statistical conditions. So, which is exactly what we are trying to do. Although most of the stuff is done by someone else, we are just going to use a third-party library to make our lives easy in this case. Again, I already summarized the cell types can be found in multiple tissue types, and similarly, a tissue type may contain multiple cell types. So now I hope you understand the difference between these two. I just wanted to highlight this for those of us who don't live and breathe biology every day. Okay, now for our work, we are going to use a library called SCSA, which apparently stands for Single Cell Specificity Analysis, and this is for cell type annotation, right? So it allows for accurate cell type annotation. How does it do that? It compares the scRNA-seq data, which we have from our last exercise, or going to do it again very quickly today. And they compare those against a reference cell type profile. So they have a reference database of all cell type profiles, and this is how it's going to work. How does it actually assign it? It calculates a specificity score for each cell type, and it measures the likelihood of a cell that we have, belonging to a specific cell type based on the gene profile, right? So this is how it's working. And if you want to learn more details, again, I'll show you quickly a few screenshots of this paper that they published, and you can go ahead and read exactly how it works. But I'll try to summarize it at a high level. It includes the library that they provide, pre-built reference database for various organisms, and obviously enabling us to perform this annotation. And you can create your own custom reference database if the one that they provide is not useful for you, or not sufficient for you, I should say. So here is the paper. You can go ahead and Google search for this. It's pretty easy. They have the link on to this one even on their GitHub page. If you just Google search for SCSA, I'll leave all the links down below in the description anyway. So I underlined the key aspects that I want you to focus on in each of these next 3 screens or so. So first thing first, how does this work? It's based on a score annotation model combining differently expressed genes, right? We already identified differently expressed genes in our last exercise. So it's going to take that, and then it combines that and the confidence level of the cell markers for both the known and user defined information. What does that mean? Let's go. So the input of SCSA is a differently expressed gene cluster matrix. We have it, right, from our last exercise. In the exercise, we're going to do that. And then it identifies the marker genes of each cell cluster. We kind of identified that in ourselves in the last tutorial, so we can actually export the data right away, or import the data right away into SCSA. Now, for each of these clusters that we're trying to identify, a marker gene identification vector is generated, because it's not just one gene, right? It's a collection of genes. And it uses cell markers from 2 public data sets, databases, cell marker database and the Cancer SCA. But in addition, you can also create your own database. But if you want more information about what's included in the database, again, obviously all that information is available. You can go ahead and get that information yourself. But let me quickly, this image actually summarizes the process. So on the left-hand side, as you see, when we start our journey, you have your gene expression data, and then you extract your differently expressed genes from this gene expression, and then you kind of have a cluster where each cluster represents a different cell type, right? So that's our assumption. So for each cluster in this in multiple databases, in each of your databases, a cell gene matrix, let me use my mouse here so you can follow, where is my mouse, so you can follow the what I'm reading. For each cluster, each database, a cell gene matrix with 2 vectors will be generated to form a raw score. So you can see a raw score is formed right there. So you have your cell gene matrix right there, and ELL are the 2 vectors that are generated to form this raw score, what they call S1. Now, if you are S right there, 1 for the cluster number 1, for example, and if you have multiple databases that are selected, then vectors would be normalized. So when you combine that, you actually end up with a new vector called Z, labeled Z in here, and then you multiply that with certain weight right there, database weight matrix, to make the last uniform vector. And in the last step, these vectors, the ranked cell type vector, will be generated according to this uniform score. I know that was a very fast, quick summary of what's going on. If you actually go to their page, they even have math for you, for those of you who absolutely love to look at these math equations, and that's how you learn how things actually work. But eventually you have this pipeline, and they also add something called Go enrichment analysis to give some clue on unidentified clusters, because there may be some unidentified clusters. So if you want to get some information, it also performs the Go enrichment analysis. And then eventually what comes out is, okay, for cell type, for cluster 1, here is the cell type, and here is the score. And I think it reports like the top n cell types that it identified, and then it gives you the scores for each of those. Then you as an expert can actually accept, okay, I'll just take the top one from each of these, or you kind of need to evaluate if that makes any biological sense or not based on what cell type it's actually reporting. Okay, I think that is a quick background. I'm sure all of you are eager to jump into the code, so let's go ahead and start looking at our code. As usual, I'll share the code with you. Look for the link down below. And this is basically, I built this on top of what I've done in the last tutorial. So if you have watched that, you can kind of fast forward a few minutes directly to get to the point. But for those of you who did not watch the last one, here's a quick overview. Dataset downloaded from this link. Luckily, 10x actually made multiple data sets available for us. This is one of those, and this is mouse brain. And this is a data set that has been collected using one of the 10x's products called Visium. And I briefly summarized what that is in my previous tutorial. So please go ahead and watch that video. And the primary library we are going to use for today is ScanPy. As you probably know, if you're working in Python, for R, there are multiple packages and or equivalent, I should say, in R. So I installed our ScanPy, and this is Google Colab. And I also installed ANData right there because we are going to work with annotated data. And again, all the explanation here, I'm going to share this, so go ahead and read that. Imported all the required libraries, and then I am importing this one data set from these data sets that they shared, and I'm loading the H5. Again, you can load Visium data, you can load any other types of data using your ScanPy. In this example, we are obviously working with Visium data, and H5 is the file that I'm going to load from this location. So, okay, right there. And then immediately it says, hey, go warning, variable names are not unique, blah, blah, blah. To make them unique, go ahead and run that, which is exactly what I've done. Variables are your gene names, and observations are your cells. So number of observations we have in this data set, 2,987. Number of genes, 31,000. Almost all the genes that are out there are present in this specific data set. So this is how we started off with. And our ANData container actually has, our object actually has these elements like observations, variables. You have the two spatial, like right here. And observations is this data within the tissue, and so on. I know I'm repeating what I've done in the last video, so let me kind of slowly go through. Now, if you look at the observations, you can see how this, how the cell, each specific cell, or in this case spot or location, you can call it, is barcoded right there, and is that within the tissue, and where exactly is that within within your spatial coordinates, are these located? Okay, and if you look at the variables, again, you have 31,000 such variables, and each of these, and you can see each gene named right there. And the next one is what we were looking at, what is the highest expressed gene. And you see mitochondrial, the top highest expressed genes are all mitochondrial genes right there. And we are like, okay, MTCO3 sounds interesting. We'll probably have a look at it spatially in a minute. And but the ANData object also has X, where it's basically a sparse matrix that contains the for this data set, and you can go ahead and print it out. Quick side note right there. Okay, now we did a few investigations. Basically, okay, what are all the genes with non-zero expression in a given cell? So you identify one specific cell and just show me all the genes with non-zero expression, and you can just go ahead and print that out. So at this point, it's just a matter of what exactly are you after. in this specific data set. But since you collected this with the spatial reference, right? I mean, transcriptomics with the spatial reference, you probably want to do something in association with the spatial location. So it makes sense for us to go ahead and visualize this. This is where all the data came from. And please note that for Visium, each data point right there is not a single cell. It's actually probably 3, 4, 5 cells, because this data point has certain dimensions and multiple cells may be under each of each spot right here. But still, it gives us pretty nice idea about the spatial information, from this tissue. If you only want to have a quick look at the tissue itself, you can see the tissue looks like H&E stained, and you have all the nuclei showing up right there distributed. Okay, so that's quick information. And then we did a few other things like, okay, I am interested in a few specific genes like MTCO3, for example, that we identified as the one that shows up a lot. Where is it located? It's pretty practically located pretty much everywhere. So maybe not a good one for differentially expressed genes, but maybe VIP or RORB are good ones for differentially expressed. As you can see, they're kind of spread over, right, a specific part of this mouse brain. Moving down, now we started cleaning up of the analysis, and normally if you are not interested in anything, go ahead and calculate the QC metrics. But if you care about, for example, mitochondrial genes, then it does make sense to add an extra column to your AND-data object where you add a tag of, is this mitochondrial, is this non-mitochondrial? Because we do identify 13 different mitochondrial genes in our data set right there. And once we learned that, okay, we do have enough of these and you're interested in this mitochondrial or statistics of these mitochondrial genes, then you can go ahead and run this line to actually add this column where you say, okay, is this a mitochondrial or not? In this case, obviously, none of the ones that you see on the screen are mitochondrial, so they're all false. For the ones that start with lowercase mt, of course, it would change that to true. Lowercase mt because this is a mouse brain, so lowercase mt, uppercase mt for a human brain. And in the last exercise, we also added some ribo-based genes and not just mitochondrial genes. But for now, I just commented those lines off. Let's just keep things simple. And then we did calculate QC metrics. Now, when you look at the AND data object, then it's not just a few pieces of information. It actually added all the QC metrics information because it performed the log 1p and number of genes counts, and it also did log 1p of total counts. And also when you look at the variable, like for the genes itself, it did add this MT. We already did that, but it also does added these number of cells by count, mean counts, and log of these mean counts, and so on. So now we have good idea of all the statistics that we kind of need to understand our data better. Okay, now let's go ahead and start investigating. That's what we did. Okay, what are the number of spots and how many mean reads per, some high-level statistics, which we obtained right there. And you can visualize the result, the table that we have here. And for example, if you look at mean counts or number of cells by counts, for example, this specific gene actually is in 0 cells. So probably not useful. In 0 cells right there, probably not useful. And also percent dropout by counts, 100% dropout, 99.9%. These are all like low quality, but if you look at this 48%, like the lower the number, the better it is, right? The higher the number, the higher dropout. So maybe we can filter them out by either total number of cells by count or dropout rate. You have multiple ways of filtering the data. In this case, we actually chose to also look at observations and see how else can we filter that out. You can see the total counts. So this specific cell has that many total counts, for example. So you can say, okay, if the total counts are below certain level or above certain level, I'm not interested in it. So that's another way of filtering. So once we understand that, we went down and we said, okay, let's also have a quick look at our how the data is spread, like total counts. Like you have some of these cells with very a large number of counts and some low counts, and same with number of genes by counts. Maybe this gives you an idea of where to set your limits. Yeah, again, visually looking at it. Now we are sorting the values based on total counts, and you can see these are 3 counts, 43 counts, and so on. So you can, this again may another way of looking at your data, so you figure out exactly how to filter that out. My personal preference is actually plot these violin plots. That kind of gives you exactly where bulk of your data lies and where the outliers are. So you can kind of set a threshold. In this case, I don't know, somewhere around 6,000, somewhere around 2,000. So this is the data I want to work with. Also, from the total counts point of view, maybe I'll set a limit at 25,000-ish or 30,000 and then only work with this data. Also, if you are interested in the mitochondrial genes, then go ahead and set a limit around between 20 to 30%, obviously. So that's exactly what we have done here. So we said our minimum counts are 5,000, maximum 35,000. And also the percent mitochondria less than 20%, meaning all of these ones. And if you have ribo, then you can add some additional conditions over there. And then we plotted, and now this is the data we are working with going forward. Okay, even that one, if you want to filter it out, then you can basically say minimum number of cells for each of these genes must be at least 10. Otherwise, I don't care about this specific gene, right? So this is another way of filtering, additional way of filtering. And now you have the data all cleaned up, and have a quick look. Now the data hopefully looks good to your liking. And now let's go ahead and perform some other analysis. In this case, let's find the gene with most counts and plot it before and after normalization. So now we have started to perform the normalization. So here is our MTCO3. Although that's not of interest for us, it's a good one where you have lots of counts, so you can kind of see how the data is changing when you're performing certain actions. For example, in this case, normalization. So the data, the values are between 200 to 1200, and then you perform the normalization, and you can see that, okay, now the spread went between 150 to 450. Why are we doing normalization? Because we have so many genes that are expressed in different ways with different intensities and so on, and we have different cells that are also so diverse. We want to bring everything into a manageable range. So any outliers, for example, are not severely affecting whatever you're trying to do. So a good way of doing that is obviously normalizing and then applying a log onto it, so the range becomes even more narrow, so it makes it easy for you to work with. So that's exactly what we have done. We perform log transformation, so this is where you are. And then now we are ready for identifying your highly variable genes. And for that, we did, of course, as part of ScanPy, highly variable genes right there. We used a Seurat approach where we said, okay, just show me the top 2,000 genes. Top 2,000 highly variable genes, and that's exactly what we have down here. And if you want to visually see, this is your total data, and these are the highly variable 2,000 ones that you are using. Again, these are we are still filtering right here, but after normalization and log transformation now. Okay, now that we have our highly variable genes, let's go ahead and take only the top 2,000 highly variable ones to work with going forward. So now that we have this 2,000 genes, that's too many dimensions for us to work with. Let's figure out, okay, is there, can we reduce this dimensionality? And we performed principal component analysis. And if you don't provide any additional parameters there, by default, it's going to calculate the top 30 principal components. Usually the top 10, you'll have like all the 90+% of the information stored in the top 10. So you can drop the remaining. That's what we are trying to figure out here. So what is the variance ratio right there? And if you look at the top 10 to 12, where this thing kind of elbows out, this is where you have maximum information, and here you don't have as much information, meaning we can afford to lose these components. So that's what we did. Okay, let's go ahead and use only the top 20 in this example. And then using those 20, we want to further reduce the dimensions to 2 or 3 dimensions. In this example, we chose to reduce the dimensions to 2. And for that, we used an approach, UMAP approach right there, Uniform Manifold Approximation Projection. So that's what we did here. And there you go. So this is, after all that, our genes, our data is mapped in these 2 arbitrary coordinates. I say arbitrary because you cannot kind of make biological sense out of this directly by looking at UMAP1 and UMAP2, but you can clearly see that, hey, I have like various clusters here. Now you can choose to divide these into 1, 2, 3, 4, 5, 6 clusters, or you can choose to say, hey, this is a different cluster, 1, 2, 3, 4, 5, 6, 7, 8, 9. You can divide this into infinity. Well, you can divide this into how many ever dots you have, right? So what is the right balance? That you have to experiment in an iterative way. But how do you control that? Well, in a way, first thing first, let's go ahead and visualize this. And here is for a specific gene. For example, if you're looking at FTH1, most of the counts for FTH1 are coming from this part of the cluster. So this cluster probably represents an area in spatially where FTH1 is expressed higher compared to other regions. Same with whatever this other gene is, right? So this is one way of understanding exactly where what these different clusters actually mean. But at each of these data points, it's not just FTH1, right? You have multiple genes located, and this is the profile we need to understand so we can label this cluster appropriately as some type of a cell type. We're getting there slowly, as you can see. And then after this, we wanted to perform cluster analysis, like separate them into individual clusters. You can use k-means, for example, but it's very customary to use a Leiden algorithm. So that's what we installed right there. And then we applied certain resolution. Again, changing the resolution changes the number of clusters. So go ahead and play with this one. And with a setting of 0.6 with my data, I think I got 13 clusters. With a setting of 0.4, I got 8 clusters. So you get the point. You get the idea here. Okay, so when you run that, what happens is to your A data, if I scroll this all the way to the right, if I can find the bar, yeah, right there, you should see that a new column got added that's called clusters. And it has number 0, 5, 1, 6. So that basically labeled each cluster a name, 0, 1, 5, 6, whatever that thing is. Again, our goal is to add another column called cell type and say what cell type 0 corresponds to, right? Find out what cell type 0 corresponds to. Okay. So now that we have clusters, we just plotted on the screen, and there you go. These are the clusters that we have. And you can also overlay onto the actual tissue spatial locations, and you can see that, okay, all the purple dots right there, we label them as number 4, but again, we don't know what cell type that is. Now, it is possible that 4 and 5 are not different. I mean, we adjusted the sensitivity of our clustering algorithm to separate these two regions as 2 different clusters. Maybe they are 2 different tissue types, but not necessarily 2 different cell types or so. So maybe we'll start with 13 clusters here, but after performing our cell type analysis, we'll probably end up with only 5 or 6, because it may say, hey, this cell type is same as this cell type, so you have only one cluster right there. So we'll find that out, but hopefully we understand where our starting point is. Okay, now we start our finding the marker genes for each of these cell clusters. So there you go. You can go ahead and rank your gene groups and plot them. So for 0 versus the rest. So for each of these clusters versus the rest, you can actually see what the score is and what the top genes are. So you can kind of see individual genes in each of these. I know these are too small for you to see, but again, this is one way. The better way of looking at this is using a heat map. So for group 6, for example, go ahead and plot, and it tells you NRGN. You see some high signal coming right around here. So you go ahead and plot that to see exactly where this NRGN is coming spatially. Now you have much better understanding of what's going on from a spatial point of view. Okay. Now, we are almost getting to the starting point of what we want to do. I promise it's not going to be a very, very long video, but we are getting there. These steps are very important for at least for us to understand, so we know what the starting point is. So then we printed the top n genes per cluster. So we have each cluster, right? Now, what are the top, for example, 10 genes are like we are plotting out here. So for example, our cluster number 0. The top 10 genes are, cluster number 0. The top 10 genes are these ones. Now, if you are smart enough to know that, hey, these are the top 10 genes, so I know what the cell type is, I know what the tissue type is, amazing. But most people are not that way. We need to match these against certain database. So it gives us probabilities of what this, or some sort of a score of what it thinks or suggests the cell type is for that type of profile. Okay, so that's the goal. So let's go ahead and move down. Now, to start our cell type analysis, we need to export our data into, for example, a CSV file that can be imported into our next step. So that's what I have done here, exported our data set right there with these columns, names, log fold changes, scores, and p-values, right? So it's going to use this information to perform the cell type analysis in our next step. And we save that to a CSV file. If you open the CSV file, this is exactly how it looks like. For cluster number 0, this is the name. N is our name, right? So that's the name. And for cluster number 1, that is the name for that specific gene. And what is the score? 57. 56 is the score for the next one. 50 is the score for the next one, and so on. Same thing here. What is the score for each of these, and what is the p-value for each of these? Now you can set a threshold on what to accept as part of your cell annotation. You can basically say, hey, I don't want to accept anything below certain p-value, or above certain p-value, or you don't want to accept anything below certain scores. And all that can be done as part of your SCSA type of analysis. And again, there are various ways of actually going from here to cell type annotation. You can actually use machine learning if you want to be fancy. You have a known data set, you have a known database, and you have known gene profiles, and you can train a model, support vector machines, random forest, you pick it. You can train a model and you can say, okay, this is my X values, which is your gene profile. This is my Y values, which is your cell annotation or cell type. And then say, okay, I want to train it. Now you have a model. And now you can just send like this future data or unknown data, and then it gives you the probability of what your cell type is. This is not a machine learning approach. This is more a statistical approach that we are following here. Again, the links are down here and also as part of the description if you want to learn more about it. But let's now start executing some of these commands. First thing first, what is our present working directory? Again, I'm in Colab, so it's the content. Like if I go back up here, it's stored as part of this content. But once this runtime is disconnected, all that stuff is gone. So of course, you can repeat this in your local notebooks or local just the IDE, whatever you use. But just a quick warning that I'm using Colab and I'm selecting a location that will be deleted when the runtime is closed. Or you can link your Google Drive, and then you can use that as the path if you want. Okay, quick note. Now I'm cloning this repository scsa.get. It's already exists because I've already executed this command. So let's go ahead and check. So it's right there, right? Under content, I have a directory called SCSA. And remember, the file that we saved, scanpyresult.csv, is located right here with this Excel or CSV document right there. Yeah, so that's right here. Now it created an SCSA, and what does SCSA have? It's basically the repo from the location that you see here. It's got the CSV files, like for example the mouse.csv files, like the database, and whole.db is a database, actual database. That's everything. That's all of this. Or you can pick individual ones if you want. Okay, so I think that's it, and I already saved result, but let's do the exercise one more time. So we cloned it, and if you look at the documentation, it says go install OpenPyXL. So I have done that, so I already installed it, and now let's go ahead and check our present working directory. It is still /content. We need to be inside SCSA, because once you have this library, all you are doing is a command line. That's it. There's no lot of coding. You'll see that in a second. So let's go ahead and change the directory to SCSA so we can run the file. Which one? There should be a Python file here. scsa.py. So we can run this scsa.py space whatever your input data is, space whatever the other parameters is. So we are going to provide some command line arguments in a minute. So I am in there. Now, how do you run this scsa.py file? So like I just said, command line scsa.py -h, obviously help, -i is the input, o is the output, d is what type of database are you going to use. We are going to use the whole database in our case. -c is the cluster, and so blah blah blah, all that stuff. I don't want to go through the document right now, but I'll show you the line that we are going to use, which is scsa.py, right? We are doing a command line. Look at the exclamation right there, and then Python scsa.py. If you're doing this on Windows, it's going to be just Python space scsa.py, blah, 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 in the environment that you're working in, right? Okay, minus D, we are using the entire database, minus I, which is basically where is the input coming from, which is our scanpy result.csv, and minus S is the source, ours is scanpy, and you have a few others. I think let's go ahead and look at it. It can be cell ranger, Surat, it can be scanpy, it can be SCRAN, like scran. So we are using scanpy, and minus G is the type, minus G is the species, human or mouse. And in our case, of course, it is going to be a mouse, so we left it to mouse right there. We entered mouse right there. And a quick note on the p-values. We, in this example, are using a p-value of 0.02, which means that is the cutoff threshold. Anything above that is going to be eliminated by default. But remember that a low threshold, for example, anything below 0.05, it helps you in minimizing the inclusions of some of the genes that just show the differential expression by chance or in a random way. But at the same time, obviously that means reducing the risk of false positives. But at the same time, low value also means you're excluding potentially genuine markers, but showing very weak differential signal expression. So you need to play with this to see what type of effect it has on your end result and how drastically is it changing your cell types. Usually not much. If you go from, let's say, 0.02 to 0.01 to 0.03, that's a good indication of having a robust, one indication of having a robust cell type annotations. Okay, with that information, let's go ahead and run this line. And you will see stuff on your screen, start to see some results. Okay, cluster number 0, let's go back up so we can focus a bit on it. Cluster number 0, it is done analyzing it. It's done matching with the whole database, and it's like, okay, coming up with the scores and says that, okay, brush cell, it's is what cluster 0 is, with a score of 5.03 and the times right there. And the next best one is this stem cell 3.9 or so, and then it gives you all the details about the p-values, q-values, everything. We're going to dump it into a a text file, so we can all the results into a text file, so we can better easily summarize all the information. So the next one is also a brush cell, and if you keep going down, then cluster number 10 apparently is also a brush cell, and then this is a ciliated cell, and the next one is the ciliated cell, and so on. Okay, so now let's go ahead and go all the way down so we can, that's a lot of information. Okay, so now we can actually save this into our result, all of this, as a text file. It's saved as a text file. Obviously, we provided the output location as part of our command line, but now we are assigning that to a pandas data frame, so it makes it easy for us to go through the results. So let's go ahead and read that and go ahead and look at the last few values. You see how it shows like minus 0.41 z-score. That's extremely low because we are on the tail side of it. Let's go ahead and plot the head of it. So we see the top ones in our data right there. So for cluster number 0, the highest one is the brush cell with a value of 5.03. The next one is stem cell. We just looked at that data, so we know what this is, 3.96. And the third one is a spiral ganglion neuron. But if you map this onto, go back to your clusters and see where they're located, and you are a neuroscientist, you know that, hey, this cannot be brush cells or stem cells. This is actually a spiral neural ganglion because for whatever biological reasons, then of course that is what you should be assigning. But this gives you a good idea of what the top n number of z-scores are, and you can probably investigate the top 5, 6 and see. But that's a big number, like 5 compared to 3.9 compared to 2.9. So it is much more confident that this is a brush cell than anything else. But still, you have to wear your biology cap and make sure that these things are actually valid. Okay, now that we have these, we are going to group these by cluster. Well, what am I doing? I'm just trying to pick the cell types with highest z-score, because I know, okay, I have all of these, but I want to assign each cluster to the highest one. That's what I chose to do here. And now you can go ahead and print those, and now you can see, okay, these for all of our 13 clusters that we identified, the top 3 are brush cells and brush cells and the B cell cardiomyocyte. So you can see that even though I have 13 clusters, some of the cell types are pretty much the same. So now create a dictionary of cluster value like 0. So all I'm trying to do is map the zeros to each of these, so I can kind of add a column in our A data, so we can easily plot and investigate this data further. So let's go ahead and look at the cluster. So there you go. We currently have numbers 0, 5, 1, 6, 3, and while I'm coding, I was getting some weird error, and I started looking at it. Basically, these numbers are not integers, they're actually objects. You see how they are within quotations right there? So I'm just changing them to integers right here. Again, not necessary depending upon how you have done the analysis until this point. And now I am mapping the cell types to the clusters. So now when you look at the observations all the way to the end, let me scroll. Now we added to cluster 0 cell type, breast type, myocyte, and so on. Okay, that wasn't that bad, right? I mean, the first part was just going through what we have done in the last video, but these few steps are like literally pretty straightforward and easy. And now let's look at, okay, what are the unique ones? Now 1, 2, 3, 4, 5, 6. We only have like 6 types that are actually unique. Change this value, the p. Actually, let's do that. This is very important for us to, in a way, investigate this. So where is our where are we cell types, blah blah blah. I just want to show you how changing the p type may affect this information. So let's go back up. Okay, let's go up a bit faster, and yeah, right there. Sorry, let's just do 0.01. okay, 0.01, and then run this and come back. This should be pretty fast. Okay, so once this thing is done, it's just checking a database, so it should be relatively fast. So once that's done, let's see if it increases the number of clusters or number of cell types for us. Okay, so once that's done, blah blah blah, I said it's gonna be fast. Not for the video purposes, but in general that's fast. Okay, let's go ahead and look at this. Now you can see it still says brush cell for the cluster number 0. So I would be shocked if it completely changes everything, although the stem cell score kind of went up a little bit by restricting our p-value even lower, right? So now let's go ahead and look at the cell types and z-scores, and now let's look at it. So now the second one previously was brush cell. Now we changed that to osteoclast. Yeah. Now let's see how many clusters do we have. I think we end up with 8, if I am correct, right there. And let's go ahead and change those to integers, and let's go ahead and see how many of these. And let's go ahead and add it to our A data object. And now let's go ahead and look at it. So now we have 8 of these. Yeah, 1, 2, 3, 4, 5, 6, 7, 8, 9 of these. So let's go ahead and plot it onto our image and spatially, so we can understand exactly where these are located, and we'll end the video right there. I really appreciate your patience for watching all of these, and I'm pretty sure some of you probably forwarded this a little bit, but yeah. You see how it makes, you can see all the blue areas, right there, B cells, and then all the green right there is cardiomyocyte, and the red ones are ciliated cells, and so on. Now you know spatially what these means. Now you come back and say, that absolutely makes no sense. This is not a heart muscle. You cannot see cardiomyocyte right there, so it must be something else. Go back to this cluster number 1, 2, or 3, whatever, and then see that, okay, the one with the second score makes more sense. So go ahead and assign this to that specific one. But I chose to pick the top ones in all of these clusters. So this is how the output plot looks like. Okay, I think that's already a long video, and thank you very much for sticking until the end. And if you want more similar type of videos, please let me know by commenting here, and I seriously hope you find these to be useful. Thank you.